Very good. Now we'll continue with the formal introduction. Cuajungco is a professor of biological science who joined Cal State Fullerton in 2007. He earned his PhD in neuroscience from the University of Auckland, New Zealand, after doing his pre-doctoral research at Harvard Medical School investigating abnormal zinc metabolism in Alzheimer's disease. As a postdoctoral trainee at Harvard, he helped discover the gene that caused familial dysautonomy, a sensory neuropathy. He then continued his postdoctoral work at Stanford University, studying signal transduction molecules involving curing. He currently investigates the pathological processes and potential therapeutics for mucolipidosis 4, or MLIV, a neurodegenerative disease afflicting some Ashkenazi Jewish children. The MLIV disease parallels molecular biomarkers observed in Alzheimer's disease and similar age-related brain disorders. Please welcome Professor Cuajungco.
Yeah, so I have my lecturing voice. So I'm not sure. If you could not hear me, you could let me know and I will use a microphone. Otherwise, I will keep my lecturing voice kind of shouting a little bit so that everyone at the back could hear me. So thank you for having me, and thank you, Janice, for inviting me. As Janice mentioned, I'm a neuroscientist by training, and here at Cal State Fullerton, I'm a professor and I teach cell biology in addition to neurobiology. And Joyce is actually my former colleague at the biological science department. So the title of my talk is how to basically protect or shield ourselves from Alzheimer's disease. This stems from my previous work as a PhD student and a little bit during a postdoc. And what I do now is actually slightly different. It's called mucolipidosis type 4, which is, as Janice mentioned, it usually affects children of Ashkenazi Jewish descent. But there's a little bit of parallel, as we discovered in my lab, that certain markers in Alzheimer's disease is associated with the disease that we're working on right now. To give you an outline of what I will be giving today, so just a quick overview of the brain. Just to give you an idea what the brain looks like, what parts are involved in thinking, in deciding what to do, in our daily lives. I'll also mention the cells that are in the brain, what types they are, and how they're involved in sensory and motor. And also the areas that are affected by Alzheimer's disease. I'll have a few slides to talk to you about that. And then finally, I'll talk about the pathology of Alzheimer's disease. What causes Alzheimer's, at least what we know. And then I have a couple of slides that tells us about the current state of therapeutics in Alzheimer's, which is not very promising, unfortunately. And then finally, I will explain what SHIELD. SHIELD is actually an acronym. So I'll explain what SHIELD is and how we could apply SHIELD in our daily lives, and basically ending with a few remarks. And at the very end, you could ask me questions if you have some questions. All right, so a brief overview of the brain. So the brain actually, as you see here, this is a cartoon of a brain. You have what you call the cerebral cortex, and it's color-coded how the cortex is actually divided in regions, right? So the red that you see there, it's called the frontal cortex or the frontal lobe, and the bluish area is called the parietal lobe, the greenish area is called the temporal lobe, and then finally at the back, the purplish area is called the occipital lobe. So these regions have distinct functions that at least neuroscientists determined over time. The frontal lobe is actually involved in consciousness, higher thinking, cognition. The parietal lobe is mostly involved in spatial analysis, mathematics, speaking and language. Occipital lobe is involved in seeing. The fact that you could see me in front, your occipital lobe is actually being active right now. And in addition, your temporal lobe is also being activated because you can hear me. And eventually, hopefully, at the end of the day, or a week from now, or a month from now, you will remember this because your temporal lobe was super active such that your memory actually was ingrained in these brain cells within the temporal lobe. And this is how a real brain would look like. Unfortunately, the real brain does not have color codings as well. All right? So you will have to rely on certain markers of the brain in order for you to distinguish. And as neuroscientists or neuroanatomists, we actually have markers that determine which part of the brain is the front, which part is the parietal and occipital and the temporal. As you can see though, however, the temporal lobe is quite obvious. It's on the side or the lateral aspect of the brain. And the brain is actually, it consists of two hemispheres, the left hemisphere and the right hemisphere. If you slice in between these hemispheres, you'd actually see this cartoon where you can still see the cartoon has that color coding where you see the frontal cortex, the parietal, the occipital, and a little bit of the temporal cortex. In between, you have a fiber or a fibrous tissue that actually connects the left and the right. So you probably don't know that your left hemisphere actually talks to your right hemisphere and vice versa. The reason why is that in order for you to execute movement, for example, both hemispheres need to communicate. And in terms of determining spatial orientation of objects, both hemispheres also need to communicate, and as well as seeing as well. And right in the middle is basically consists of several internal structures that are involved in processing all the information that comes from the outside or the environment, and then being processed by your left and right hemispheres. So some structures here are quite important in relaying these informations from the environment and then from your brain out to your environment. So how you behave in the environment, for example, you detected that it's actually cold outside, therefore your response would be to wear a coat. Exactly. So you have to wear a coat. And that is because your brain sensed that the environment is cold, and therefore your action is now to wear a coat or put on an extra layer of clothing. And if it's hot, it's the opposite. So your response would be to take off your coat or put on something that is thin so that you don't get very hot. And similarly, this is how a brain would look like. As I mentioned, you have two hemispheres. You're looking on top. The left is on the bottom, and the right is on the top. And you'll see all these foldings. One reason why the brain is folded is because through evolution, the folding allowed for the brain to increase the surface area. And probably not many in this room know that that surface actually is where the brain cells are located, not necessarily inside or deep inside the brain. And I will show you later the types of brain cells that you will see in these surface area of the brain, or which is basically the outside of the brain itself. This is the brain as you will see it from the bottom. You will see certain structures here. This is the brainstem right here. And the spinal cord actually extends down, but in this particular picture, it was cut. This part right here is what you call your olfactory bulb and olfactory connection to the brain that allows you to smell. And if you could imagine, this is actually just above your nose. So how did we learn about brain function? Unfortunately, we learn brain function due to misfortunes of others, right? And these misfortunes could include a wide range of disorders. For example, person having stroke or having traumatic brain injury and damaging their part of the brain. This is an example where a person who actually damaged part of the frontal cortex produced an effect such that the person could not utter words anymore, right? And this region actually is quite famous. It's actually called Broca's area. So a physician called Broca actually discovered this patient, that the patient could not talk properly, although the person could still understand words, but the person could not utter the words coherently. And when the person passed away, the person donated the brain, and Dr. Broca actually looked at which part of the brain was damaged, and it so happened that this region within the frontal cortex was the area that was damaged. And therefore, this area was called Broca's area. Phineas Gage, who's heard of this fella? A few? Yeah, so Phineas Gage is actually one of the famous patients. He's a rail worker, and an accident happened when he was working on the rail, an explosion such that a huge metal bar that he's holding actually shot up under his neck, went through his face, through his brain, and luckily he survived. And unfortunately, even though he was fine after the accident, there was quite a distinct change in his behavior. And when he passed away, he basically, he donated his body, and they realized, so this is the skull of Phineas Gage. It's actually stored at Harvard Medical School. They noticed that the metal bar actually went through his brain and damaged part of his frontal cortex. And from that observation of how he changed from a normal person, meaning shy, social, to a quite obscene, person, allowed us to actually determine that the frontal cortex actually determines, or at least controls, our behavior, such that the executive part of our behavior, such that in a social context you have to behave, and you actually know that, and that frontal cortex tells you that you're supposed to behave accordingly. And when he damaged that part, he lost that control. So one way to also identify how brain function, or at least parts of the brain, is to use imaging techniques. So nowadays we have techniques called MRI, or magnetic resonance imaging, and this is an image of a person undergoing through MRI. And probably some of you have actually done this, where you actually you lie down, your head and neck is actually still, so they actually lock your head so that you could not move your head. And then you go through this machine. It's very noisy. It's actually a magnet that goes around, and that magnet allows for your brain to be imaged. And that image actually is this. So this is an example of an MRI image. This is a mid-sagittal view, as I introduced you earlier. Right in the middle, you have your cortex right there. You have your fiber. The fiber is actually called corpus callosum that connects the left and right hemisphere. This is an image called coronal section, where you're actually viewing right in front of the face. And this is an image on the top, where you're looking on top of the brain. One thing I did not mention earlier is your brain actually is hollow. There's actually space in your brain. And you will notice here that space right here, and those dark areas are actually called the ventricles. So the brain has ventricles. That ventricle is actually filled with fluid. What do you think is the reason why our brain is hollow. Any ideas?
Protection.
Yeah, exactly. So I heard some answered protection. So actually, the hollowness of the brain, where the fluid is located, is actually to serve as to protect the brain. So the brain is enclosed in sheets that protect it in addition to the skull, but there's also a fluid that protects the brain from movement or sudden movement as well. All right? And these ventricles that are filled with fluid allow for that protection. All right, as we basically enhance the techniques in imaging the brain, scientists actually develop what we call a functional MRI, functional magnetic resonance imaging. And what does functional MRI do? Well, it's similar to what you just saw. It images the brain. However, it also allows for the scientists to determine that when a certain task is done while you're being imaged, they could actually look at regions of the brain that is active, right? Or regions that are active, because if there are more than one. And these activities suggest function. So if you are moving your hand while your brain is being imaged, and let's say a part of this region on top of your skull or your brain itself is active, then that suggests function for movement, right? And this is one way to determine that certain parts of the brain actually is involved in particular action. And I have actually embedded a short video basically just to let you know what is happening in imaging and how these new techniques in imaging actually allows for us to determine the function of the brain.
Just as explorers have always sought to map new worlds, neuroscientists have sought to map the brain, trying to chronicle its connections and deduce how it works. But making a brain map is easier said than done. You can pick a particular property of the brain to base your map on, like its structure, or its connections, or cell types. Here's one early attempt, published in 1909 by Corbinian Brodmann. He noted how the cells look different from one parcel of brain to the next, and his maps are still used today. But depending on the property you look at, your maps can end up quite different. So neuroscientists can't even agree on how many areas there are, let alone what they all do. But now a team has made what could be the most accurate map yet. It's based on hundreds of brains and lots of different properties of those brains. It's even defined a whole bunch of new brain areas. Like this one, Area 55B, which the team thinks has a role in language. It's pretty obvious here in a map of language-related brain activity. And again here, when they look at the connections between this area and others. Not so clear here, though, on a map of how much insulation there is around neurons. That's why combining all these data sources is so important. It might even mean neuroscientists can finally figure out what the brain's borders and territories really look like. Explore the new map and plug in your own brain data at humanconnectome.org.
All right, so you might have heard connectome. Connectome is also one of the big areas of research right now in neuroscience, where they use imaging techniques to actually look at the connections between the left and right and how these connections actually relate to function as well. All right, the hope is that by identifying these connections, we're able to clearly dissect specific functions of brain areas. And as you see on the video, the way they image the brain and the way the brain is demarcated those lines that you see, the squiggles, are actually individual parts of the brain that are involved in specific functions. So now we are going to the cells in the brain. There are actually a lot of cell types in the brain, but the ones that are known are neurons. So the neurons typically look like this, and some look a different or oddly shaped. So within a neuron, you have what you call dendrites. And the neuron actually, as you could imagine, is like a tree that has arborizations. And these dendrites represent the arborizations. These dendrites connect with other neurons. And some brain cells are actually called glia. So glia means glue, because earlier when scientists looked at the brain, they identified these cells that appear to be sticking with neurons all the time, or always associated with neurons, and therefore they named this glia. One glia, or glial cell, that is well known is what you call an astrocyte. And if you notice here, this is a neuron, and this is an astrocyte. The astrocyte was named based on how it's shaped. As you notice, astro is synonymous to star. So the star shape is connected to the neuron. It's also connected to a blood vessel. So for function, astrocytes are actually one of the glial cells that help nourish neurons. It helps support neurons. So it plays an important function or aspect in terms of making sure that the neurons are alive and they survive. There's also a kind of glia, but it's not a real glia as such, called microglia. Microglia are small cells that is basically equivalent to the immune cells in our body. And the role of these microglial cells is basically find or seek infection and eradicate that infection. And in cases where there's cells in the brain that die, microglia actually clean up the mess. So any dead cells that are around in the brain, that's basically the microglia's job to clean it up. And another glia right here called oligodendrocytes. As you see there, oligodendrocytes also stick with neurons, but in this case, what it does, it creates membranes that wrap around a structure of the neuron called axons. So how neurons communicate, actually, as I mentioned, so they're connected via dendrites, but they're also connected via axons, which is the reddish part right here. What happens is that when a neuron decides to communicate with another neuron, it sends an electrical signal. So that electrical signal, as you could imagine, will travel. Imagine axon as your wire, and you're connected to an outlet. So the wire is insulated, and that insulation is actually due to this cell type called oligodendrocyte. Thus, making sure that the electrical signal that passes from one neuron to the other at the very end is intact, meaning it does not dissipate, it does not go away. So the electrical signals allow for the travel smoothly from one end to the other end of the neuron. And then at the very end of these neurons, you have chemicals that are released, and these chemicals are called neurotransmitters. So these neurotransmitters, you might have heard of them. Some of them are acetylcholine, dopamine, glutamate. So they're the ones that control how the other neuron that gets these transmitters will react or will interpret that signal. So in the brain, you actually have two ways how neurons will react. One is called excitatory, or exciting a neuron. The other one is called inhibitory, or inhibiting or suppressing the activity of that particular neuron. So it's either excited or inhibited. And the transmitters that are released at the end of these neurons that are attached to other neurons will govern whether they will be excited or inhibited. This is a coronal section of a brain, and if you look at a particular section, similar to a previous slide, you have a neuron here, you have your oligodendrocyte that wraps around the axon to make sure that the signal, the electrical signal that this neuron is going to send is insulated. You have your astrocytes that are also attached to the neuron, and also blood vessel, which controls what nutrients are being received by the brain. And you have microglia, which are basically sentinels that guard for any infection or to clear up debris that is in the vicinity of a neuron or within your brain. I have a short video of how neurons communicate. Oops. So these are neurons, and as you see there, these flashes are electrical signals. Those tiny bits are actually ions inside your brain. And at this particular video, it's plus, meaning it's positive, that goes inside the axons. And at the very end, as I mentioned, you have these tiny compartments that release the neurotransmitters, and these neurotransmitters will bind to what you call receptors on the surface, and these receptors will then become activated, and the neurons will then respond. And as I mentioned, either it's excitatory response or an inhibitory response.
What were the two responses?
Excitatory, meaning you're excited. You know, if you're a person, you're like moving. Or inhibited, meaning you're just standing. All right, so the neuron will either respond or not respond. And that's how the brain basically operates, these neurons. All right, so specific areas that are affected in the brain. If you look at the brain, so this is a top view, slightly slanted. And if you slice the brain horizontally, there's a structure that looks like a seahorse. And one reason why they named this structure hippocampus is because of the actual shape. So hippocampus, if you translate that in English, actually means seahorse. And the hippocampus is actually involved in learning and memory. And again, based on unfortunate events, so there's a famous patient called HM. He had epilepsy, and in order for the doctors to control his epilepsy, what they did was they actually operated on his brain, and they actually removed part of the temporal lobe, which is where the hippocampus is located. And the result, after the surgery, they cured the epilepsy, but unfortunately, he lost his memory or his ability to retain memory. He still remembered parts of his life, so that wasn't lost because that memory is actually stored elsewhere in the brain. So the hippocampus is actually involved in memory consolidation. So whatever event that happened today, happened yesterday, happened last week, last month, a year ago, that memory is actually consolidated by the hippocampus and actually push in certain parts of your brain. So a visual memory remains in the visual area, or at least close to the visual cortex, and a memory of sound should be close to where the sound is processed, which is in temporal lobe, and so on. So these memories are actually eventually, over time, are stored in various areas of the brain. And so in the case of HM, when he damaged the hippocampus, those memories were retained, but then he is not able to form new memories that he could remember. So that is the problem. And this is how we identified the hippocampus as a structure that is important in learning and memory. This is a coronal section of the brain. So if you slice the brain from top going to the bottom, that's the top and this is the bottom. The hippocampus actually looks a little bit different. It doesn't look like a seahorse now, but it looks a little bit, curved, almost like a croissant. You could imagine it as a little, croissant shape. And so, as I mentioned earlier, so the hippocampus is actually one of the areas that are affected by Alzheimer's disease. The hippocampus is involved in learning, memory, and some of the structures here, I've mentioned about the relay station. Eventually, they become affected as well, and over time, also the cortex as well. So I mentioned earlier also, if you notice on this image, the area that is kind of grayish is on the outside, and the white area is inside. So this white area is actually the fibers that are coming from the neurons, which are on the surface of your cortex, right? And as I mentioned, eventually these cortical areas are affected by Alzheimer's disease. So what is the pathology of Alzheimer's? So right now, we don't have a way to diagnose a person whether the person is actually suffering from Alzheimer's. How we diagnose Alzheimer's is usually post-mortem. So the doctors look at the brain and stain for markers, 2 hallmarks of the disease. I have a short video here just to give you an idea what happens to the brain and what the scientist actually looks at.
The Biobank at the Sun Health Research Institute in Arizona houses more than 1,000 donated human brains. The tissue is collected, processed, and distributed to medical researchers around the world. It's priceless material that will one day lead to finding the cause and the cure for everything from cancer to Alzheimer's disease. Dr. Thomas Beach heads up the Biobank.
I'm going to show you one of our demonstration brains, half a brain really.
So this is not a freshly harvested brain?
No, this brain is, it was harvested about 3 or 4 months ago, and we used half of it for research, and we keep the other half, as you see, for demonstration purposes. It's been fixed in formaldehyde, so it's completely safe to handle.
So is this an example of a healthy brain?
This was a Alzheimer's brain, actually, and you can see some of the atrophy, the gyri, which are these squiggly things here. Mm-hmm. Normally they should be so full that they press against each other, more like at the back here. Mm-hmm. But see here, there's deep valleys between the gyri, and this shows that there's atrophy or shrinkage of the brain. Mm-hmm. And this is one of the things that happens in Alzheimer's brain.
So how severe in case of Alzheimer's is this right here?
Well, normally we would grade it as mild, moderate, or severe, and on that scale I would call this moderate to severe. So it's a lot of Alzheimer's brains are moderate to severe. Occasionally you get very severe brains where this type of change, this shrinkage, is distributed all over the brain and it's more pronounced. We have the cerebellum hanging underneath here. That's this part that my fingers are on.
And this one's a healthy example?
Healthy cerebellum, because for the most part the cerebellum is not affected in Alzheimer's disease. Alzheimer's disease affects that very part of the brain that makes us human, that gives us personality and intelligence, the cerebral cortex, unfortunately. And that's what deteriorates first. Often personality changes are early in the disease. Of course, memory and behavior becomes altered because our behavior comes from the cerebral cortex.
And this is something that wouldn't show up if you did a brain scan?
A brain scan can show the shrinkage, yes. But unfortunately, in a lot of diseases, brain diseases, there's shrinkage. So the shrinkage by itself is not diagnostic of Alzheimer's disease. Mm-hmm. At present, we really have to do a biopsy or an autopsy to get a small piece of that cerebral cortex, look at it under the microscope with special stains in order to identify the plaques and tangles that are the microscopic lesions that are specific for the disease. So at present, we cannot get a specific diagnosis of Alzheimer's disease, and plus we look at a piece of that brain under the microscope.
All right, so as I mentioned, and as what you heard from the video, so unfortunately there is actually no diagnostic way or means for scientists or doctors to tell that a person suffers from Alzheimer's until after death. Yes?
Do other primates exhibit, like gorillas, large primates, do they exhibit the same things that we have?
That's a good question.
Question, please.
So do other primates, for example, gorillas, exhibit a similar, symptoms that humans have? So in primates, they found, also in monkeys, they found that these hallmarks actually accumulate in the brains of these primates as well. And there's the hallmarks, which I will tell you later. So there are protein aggregates in the brain, which my next slide is basically, so here you go. So certain parts of the Alzheimer's brain, you have these proteins that accumulate, and yes, these primates do accumulate these hallmarks as well. And there's a research, I believe, that actually showed that some of them actually show dementia as well. So very similar. So this is an example of a brain. Again, it's a coronal section. It's cut from top to bottom, where on the right, you have what you call a control, a person that did not suffer from Alzheimer's disease. And on the left, a person that suffered from Alzheimer's disease. If you look at the ventricle with the person who suffered from Alzheimer's is very large. And if you look at the hippocampus, so this is the hippocampus right here. And you see that the hippocampus with this person who suffered from Alzheimer's is almost gone. And you also see ventricles that are enlarged in this area. And if you look at the thickness of the cortex itself, compared to the one suffering from Alzheimer's disease, it's also thinner compared to normal control. Right, so the hallmarks. So in Alzheimer's, there are actually two key proteins that accumulate in the brain. One, so it's demarcated by this red dotted line. So this is called beta amyloid plaques or senile plaques. And the other hallmark is called neurofibrillary tangle, or NFT. So this is basically a protein called tau that creates tangles inside the neurons. And when you stain them, they actually show as these black neurons or black-shaped cells. The one on the right is, again, a tangle. So this is a blow-up version of what you see on the left. And C on this panel is a slightly blow-up version of an amyloid plaque as well.
Is the tangle due to the collapse of the little tubules?
That's right. So a gentleman here actually knows a little bit about the mechanism how these tangles are created. So inside the neuron, you have actually a network of railroads called microtubules. And the tau protein is actually a component of that network of railroad. So what happens in Alzheimer's disease is that this tau, however, is abnormal. So phosphate is actually an addition, a chemical addition to the protein. And in cases of Alzheimer's, tau becomes what we call hyperphosphorylated, or there's too much phosphate that is being added into this protein, such that the railroad that is typically normal and nice and straight become twisted and tangled, and these networks of railroads now inside the neuron do not work properly. All right? Why are these railroads important? Well, inside the cell, these microtubules, or what I call the railroad, are actually used by compartments to transport materials from one end of the cell to another end of the cell. So imagine if you're traveling, let's say you want to go to, from Orange County to San Diego, and the railroad is not, the tracks are not working, then you can't travel from Orange County to San Diego. And that's, in essence, a similar situation that happens inside the brain. If a cargo coming from the middle part of the neuron cannot go from the middle to the end part of the neuron, then therefore the neuron basically has problems in transporting materials inside itself. The amyloid beta plaques, however, is not actually inside, and as you see there, it's outside the cell. So what happens is that the protein called amyloid precursor protein, it's a very large protein, gets cut or cleaved. And I'll show you a little cartoon. So this particular image, so imagine this line right here is the membrane of the cell. This protein called APP or amyloid precursor protein is actually sticking out of the cell. So let's focus right here in the middle. So this is a normal APP. There are enzymes that cut it, all right? The enzymes are called alpha secretase, and it actually cuts that little white dot. And when alpha secretase cuts this protein, it produces what you call a soluble APP, which is the alpha component, and a fragment. This fragment that is stuck on the membrane is further cut by another enzyme called gamma secretase. So they named it alpha, beta, gamma. And the gamma secretase produces another fragment called P3 or peptide 3 and AICD or amyloid intracellular domain. All right? So in a normal situation, this is the ideal way how you would want your APP to be cut. All right? Because it does not produce the beta amyloid. However, if we go on to the right part of this image, so again, you have your normal amyloid precursor protein, very long, and in certain situation, an enzyme called beta secretase cuts it. And that cut happens right above here, where the white dot is. So now it produces this APP soluble beta fragment. And similarly, it has this part of the protein that is still stuck on the membrane. However, when the gamma secretase cuts that membrane, it produces, instead of a P3, it actually now produces an amyloid beta peptide. And again, similarly, the amyloid intracellular domain, or AICD. And this amyloid beta peptide is the one that accumulates on the environment or the vicinity of neurons. And these accumulations create these plaques or aggregates. So it is actually not known how these plaques kill the neurons. But there are several hypotheses that's been provided by neuroscientists. So they say that these plaques actually produce oxygen species, reactive oxygen species. So you've probably heard of free radicals or free radical of aging. Who's heard of free radical aging theory? So free radicals are oxygen molecules that basically steal electrons from other molecules. And the free radical theory of aging is that because of these free radicals stealing electrons, this is how we age, right? In the case of Alzheimer's disease, when these plaques accumulate in the vicinity of neurons, they propose that the plaques themselves actually produce these free radicals. And now that you have these free radicals, they actually could damage the membrane of the neurons such that it becomes unstable, it creates holes, and now all these materials that are not needed by the neurons go inside and eventually kill the neurons. My former research actually implicated zinc as one of those some researchers implicated aluminum. We say aluminium. So aluminum's been implicated, iron and copper. So some of these metals actually been implicated in killing these neurons when these plaques are formed in the vicinity or the environment of the neurons.
Sorry.
Yes?
You know, they prescribe zinc and so
Yes.
forth as a nutrient and so forth, but, you know, you're saying it's bad, but I'm
Yeah, so as part of my research, when I was doing my PhD, in the end, I actually concluded that zinc is a double-edged sword. So zinc is a very important nutrient. So our body needs zinc along with copper, with iron, and some other trace metals. However, in the brain, when these free radicals start attacking the membrane, the zinc that is in the environment and some trace metals like copper and iron could actually get inside the cell and wreak havoc inside the cell. So this is where the other edge of the zinc aspect or the iron hypothesis of causing Alzheimer's or the copper. So unfortunately, we have no way of controlling how these metals could get in once the membrane is not intact anymore. But ongoing research is being done. They actually, my former mentor actually is using what you call drug compounds called chelator. So metal chelator are actually drugs that actually mop up these metals. They bind metals and they mop it up, and therefore now instead of these metals going inside and creating havoc, they're just bound by the drugs themselves.
That EDTA?
Yes. EDTA is one of those chelators. But EDTA cannot go past your brain. And this is what, so I don't know if you recall.
It can't cross the brain barrier.
Yeah, that's right. So if you recall, I showed you a picture of astrocytes stuck onto the blood vessel. So the role of astrocytes in nourishing neurons also, it plays a role in protecting neurons in having some of these molecules that are unwanted. So it actually protects. And this is what our gentleman right here in front mentioned as the blood-brain barrier. So we actually evolved to make sure that our brain is protected. We have a skull to protect. We have these sheets that protect the brain. We have the fluid within the ventricle to protect the brain as well, to cushion it. But we also have what we call the blood-brain barrier to make sure that anything that we eat, molecules that are needed by the brain, is actually prevented from getting to the brain that's not needed. And that's what we call the blood-brain barrier.
But alcohol can get through.
Unfortunately, or maybe fortunately, because of the chemical structure of alcohol allows for it to pass through the membrane. And therefore, when you have a red wine, a glass of wine, actually, I only need one glass, and I could actually feel the effects of alcohol. So, but obviously some people, based on their genetics and based on their, their physical attributes, it takes them several glasses. But anyway, so the amyloid senile plaques, it's actually called the amyloid hypothesis of Alzheimer's disease. It's suggested that that's how they kill neurons, creating oxidative stress, and the accumulation of plaques within the environment itself triggers inflammation. So do you remember the glial cell that I mentioned that is the sentinel in the brain? Does anyone remember the name? Tiny ones? Microglia. So unfortunately, and fortunately as well, because it shows you that the immune system of the brain works. So once these amyloid plaques accumulate, the microglia do its job. They actually surround the plaques and try to get rid of it. But the plaques are so aggregated that they cannot dissociate these plaques. And as a bystander effect, the problem, when the microglia try to get rid of the plaques, they produce these free radicals. And these free radicals themselves unfortunately affect the neurons that are within the vicinity. And therefore, again, having that issue of destabilizing the membrane of the neurons and eventually could kill the neurons themselves. So what causes Alzheimer's? There are actually known genetic causes of Alzheimer's disease. Right, so one of the causes, I mentioned to you the APP, the amyloid precursor protein, based on genetic studies. So this is what you call a haplotype or linkage study, where they look at family members. So you have the mother and the father. One is affected, the red shaded area, and then they look at their offspring, and the offspring is affected, and also look at the offspring. What they do is they take their DNA samples, they map these DNA, and the shaded area is basically signifying disease. So out of the map, so they have what you call a marker. So between the gene, and this gene is actually here, the APP gene. And based on this analysis, they identified that this gene is actually involved in the disease. Then what they can do now is sequence the whole gene. And when they sequence it and they look at the amino acid, so these letters that you see are actually amino acid of the protein. And the arrows that you see there, so the black letters on top are normal amino acids. If there's a mutation or a change in amino acid, it's indicated by the red. So for example here, from a K amino acid to an N amino acid, from an M amino acid to an L amino acid. So these are the mutations that have been found in the APP gene. That's just one of the genes that actually causes Alzheimer's. And as you see there, the effect of these change in the amino acids make the APP create more amyloid beta peptide. And one of the most famous mutation is called the APP Swedish mutation, because it was found in Sweden, a family in Sweden. And there's all
neurons are in the gene, and it must be arranged from one to another.
Well, the neurons express these genes as proteins, but if you have these mutations in the protein, and particularly the ones that I showed you that are red, the neurons actually tend to produce more of the amyloid beta.
Well, I asked with how many genes, how many neurons in a gene?
Oh, well, the neurons in your brain produce all these, for example, the APP.
To define a gene, it has to have a certain number of neurons, right?
Well, certain neurons produce particular genes, meaning protein, certain subtypes, and it appears that these pyramidal cells in the cortex and pyramidal cells in the hippocampus themselves produce the APP that make the hippocampus and cells from the cortex susceptible to Alzheimer's disease. So certain parts, as you see, some parts of the brain, like the cerebellum, if you recall from the video, the cerebellum is actually a structure in the brain that is not affected by the disease because they have different types of neurons. So depending on the neuronal type, the brain cell type, you could have a mutation that affects these neurons more, or they are more susceptible to the mutation compared to other brain cell types. This is another, so do you remember the enzymes, alpha, beta, and gamma secretases? So these enzymes, over time, they first discovered the APP mutation, and over time, they also discovered that some of these enzymes also have mutations. All right, and so let's focus right here on the left first. So this is what you call a blot, where you blot the peptide and the protein. So on top it says A beta, so that's the amyloid peptide, so the fragment of APP. In the middle is called the APP, and here it's called the APP SWE for Swedish. So I mentioned to you the famous mutation called APP Swedish mutation. So a normal A beta you will see there, on the A beta lane. And then within the APP lane, as we all know, I showed you earlier, APP gets cut and it produces an amyloid beta fragment and a P3 fragment. All right? And if you look at the APP SWE, or the Swedish mutation, so you have your normal APP band. However, if you compare the darkness of this A beta compared to a normal APP and also the P3, is there a difference that you could see? So the APP Swedish mutation produces relatively more amyloid beta and less P3 fragment compared to the normal. So the normal usually produces a lot of P3 fragment and less amyloid beta. But when you have a mutation called the APP Swedish mutation, it tends to produce more of these amyloid beta fragment, which is the bad part of the peptide. And right in the middle here, where they did an experiment, so we know that there's a mutation called APP SWE, or Swedish mutation. And I mentioned to you the enzyme, so gamma secretase, is actually composed of subunits of different proteins. And one of those subunits is called presenilin, and in this case it's called presenilin 1, PS1. And here it's called presenilin 2, or PS2. So when they looked at how much amyloid beta is produced when there's an APP Swedish mutation, and there's a mutation in presenilin 1, and there's a mutation in presenilin 2, versus a control when everything is normal, they found that amyloid beta is a lot in the Swedish mutation, the APP. Presenilin 1 mutation produces a lot of amyloid beta, as well as a lot of amyloid beta in the presenilin 2 mutation. So this is a cartoon of presenilin, and those dots, the red dots that you see, are the potential amino acids where you could have mutation in the presenilin 1 or presenilin 2 genes. So we all know what tau is. I mentioned to you tau is a component and one of the hallmarks of Alzheimer's. Some patients actually have mutations in the tau gene as well. And when there's a mutation, this is an example where, so they used an animal model where they created a mouse with the APP Swedish mutation and a mouse with a tau mutation called P301L. So this is actually amino acid P at number 301 that changed to L amino acid. And what they found that the animals produce lots of these tangles inside the neurons. Whereas if you look at just the mutation in tau, it does produce tangles in neurons, but not a lot. So a combined effect of two mutations, an APP mutation and a tau mutation, produce a severe, what you call phenotype or outcome in the brain of these mice. So in addition to genetic risks, so the actual, genetic cause, there's actually genetic risks as well. So genetic risks, one of those genetic risks is called the apolipoprotein E, or ApoE gene susceptibility. So this is an example of plaques in the brain where they use an antibody, so another molecule, another protein that will detect the plaques for ApoE, or ApoE that is present in plaques. And that's what they found. And they found that ApoE is also associated with plaques that contain amyloid beta, which is indicated here by the anti-amyloid beta picture. So in terms of susceptibility, this is probably more relevant right here. So if you look at this particular graph, so let's first look at no E4 allele. So I need to quickly explain to you what allele means. So genes. So we know genes. We have genes. Genes produce protein. But we actually have versions of genes. So when we say version, and that's how we term as allele. How does that happen? Well, when a sperm and an egg combine, so the sperm has it has the same gene, but it has a different allele from your father. and from your mother you have an egg, you have the same gene, but it has a different version or allele. So now when the sperm and egg combine, you have the same genes, but one came from your father, one came from your mother. And those are the same genes, but it has different alleles. All right? So alleles, in this case, we have an APO or ApoE gene, but it could have different alleles that come from your father or from your mother. So on top, if you have an E4 allele, no E4 allele, which is this part right here, your susceptibility, on the x-axis is the age of onset of Alzheimer's disease. So if you trace a person without the E4 allele, the susceptibility of this person in developing Alzheimer's is not basically until later on, if this person is going to develop the disease. However, if you look at a person that has one of the E4 allele, so again, we have two alleles, one from your father and one from your mother. So if your ApoE has an E4 allele in one, so let's say my left little finger right here, and if you trace the susceptibility of this person in developing Alzheimer's over time, so if you'll notice, it's slightly less than a person without an E4 allele. So meaning this person will develop earlier compared to somebody who does not have an E4 allele. However, look at this. If you unfortunately happen to have the two alleles of E4, the one that came from your father, you have an E4, and another E4 from your mother, if you trace that, which is the red line right here, if you keep going down, it's actually even earlier. So this person who has two E4 alleles is more likely to develop Alzheimer's earlier compared to a person who only has one, or compared to even a person who does not have that E4 allele.
That's called early onset, isn't it?
No.
Excuse me, Stan, could you hold your question
So-
until let's get through?
Oh, it's okay. So this is actually late onset Alzheimer's disease. So the genetic risk factor is a late onset, and the risk factor is late onset, but the genetic cause is actually early Alzheimer's disease. So these patients develop Alzheimer's in their 40s and 50s. And this is because of the genetic cause of that Alzheimer's disease. Another risk factor that's fairly recent, so when I say recent, maybe almost 10 years ago, there's a gene called TREM2. So remember microglia? So remember microglia is the sentinel that helps clear up things in the brain. So there's this gene, which is a protein that is stuck on the membranes of these microglia. And unfortunately, if there's a variant, so that little star right there indicates an amino acid that changed from, if you compare the normal population. So that change actually makes the microglia less likely to clear up the gunk in the brain. All right? And because it's less likely, therefore, there's going to be more inflammatory response. There's more inflammation. And because there's more inflammation, I mentioned to you about the free radicals. So the ROS is short for reactive oxygen species, which is basically the free radicals. And because of that, therefore, now there's more increased amyloid beta accumulation in the brain. And again, the vicious cycle of these inflammation and free radical production, and therefore the neurons within the vicinity become collateral damage, and they die. So this is another genetic risk factor. So what's the current state of therapy in Alzheimer's? And despite the efforts that neuroscientists have done in the past 15, 20 years now, the clinical trials that target amyloid beta failed. Clinical trials that target the tau, the neurofibrillary tangles that accumulate inside the neurons, also failed. So we had major setbacks in the past many years, and this just came out yesterday, or a few days ago. Another one bites the dust, unfortunately. So another trial failed. So when I wrote my synopsis, my abstract for this talk, they found some significance. It's not great. They found some significance improvement in patients, and so this is the drug. But again, a few days ago, now they said, unfortunately, again, this does not work. This particular drug. So most of the drugs that target these amyloid plaques are antibodies. So I mentioned to you antibodies. Antibodies bind to proteins. And once the antibodies bind, your immune cell actually detect it and realize, okay, there's an antibody, this must be cleared. And the microglia will then clear it up and, again, get rid of whatever gunk that is within your brain. Unfortunately, these antibodies did not work. So when I say did not work, they do find the plaques, and they do clear somehow the plaques from the brain, but the cognitive effects, the loss of memory, problems in learning are still there. They still exist. So they did not prevent the disease itself. And that's where the failure is. So what is the next strategy? So neuroscientists are now saying, maybe we should target them even before the symptoms occur. So that's what's currently what neuroscientists are trying to do. They also did some studies in the past where people who what they called the mild cognitively impaired, or MCI people, where they gave these drugs. It also failed, and because of that, again, learning from the lessons of the failures, they're actually even saying that perhaps targeting is even before any symptoms show, so even any signs. So what they're trying to do now for those people who have genetic risks and genetic costs, so without the actual symptom, they're now giving the drugs to these people. And the hope is that early on, even before these plaques start accumulating, or at least a lot of them accumulate, that these drugs may help and produce the desired outcome. Yeah, I know it's a little bit sobering and depressing to know that as of now, there's still no drug that is effective. Yes.
How long does it take from the time that they start to the time that they determine?
The clinical trials. So luckily, Congress actually they budgeted lots of money for Alzheimer's research, and the drug trial itself takes about 10 years. All right? So when they do initially what they call the preclinical studies, they use the mouse model. Unfortunately, if they go into human clinical trials because they found it works in mice. But the problem is, what works in mice does not necessarily work in humans. So, and this is also one reason why we had all these failures, is because scientists identified the drug that works in these mouse model of Alzheimer's disease, but then when they now translate it into human clinical trials, they do not see that significant effect. So, what is SHIELD? And how can we apply it? So SHIELD is actually an acronym. And as you see there, S stands for sleep, H for handle stress, I for interact or socialize, E for exercise, L for learning new things, and D for diet. And this was actually coined by Chopra and Tanzi. So Rudy Tanzi, right here, I put a picture. He's my former director when I was a PhD student at Harvard Medical School. And he coined that along with Deepak Chopra. And they actually they have a book. So I'm not here to promote his book, but if you're interested, it's something that you could read and basically learn from what they're trying to propose. So I guess my role here is to tell you what SHIELD is about and how we could basically use this acronym to kind of improve our lives and hopefully prevent us from getting Alzheimer's disease. All right, so sleep. Sleep, it's very important. And there's actually research that says sleep is, basically a a miracle drug for us from preventing Alzheimer's. So what is the evidence? So they actually use animal models. They use actually animal models, rodent models of Alzheimer's, and they showed that these animals, when they sleep longer compared to the one that they actually experimentally disrupt their sleep, the amyloid beta protein in the tau, those accumulations are actually less, and they have better memory compared to the mice with disrupted sleep. And I have this like little cartoon here. So this is actually a recent research that came out a few days ago. So this is a cartoon of an experiment that they did. So, and they also looked at humans, how when there's a regular sleep-wake cycle in these animals, in the mouse model, so there's this kind of cartoon is color-coded. So you have what you call the inflammatory astrocytes, those reds. You have the normal astrocytes, those kind of green star looking. And you have the amyloid aggregates, the black one, the tau aggregates, the blue one, and just a simple strand of amyloid beta and then a tau. So in the normal animals where they receive the regular sleep-wake cycle, these fragments of the amyloid beta and the tau are actually cleared. So they go through the vesicles, the fluid, and then it gets filtered out of the brain, and therefore now there's less of these plaques. However, in a sleep-deprived animal model of Alzheimer's, they actually found that the neurons release these fragments, and the fragments do not get cleared. Because they're not cleared, they just hang around in the vicinity, and eventually they kind of bump into each other and become plaques, right? And the tau themselves also become plaques within the vicinity, in addition to being tangled inside the neuron. So this is a recent research that actually shows that sleep is indeed very important, and it does help, at least based on these research right now, that it could actually help minimize us from developing or getting Alzheimer's. Yes?
Parents of young children or children of any age are notoriously sleep deprived.
That's right.
Is there any difference in the onset of Alzheimer's between people who have had children and people who have not had?
That's actually a good question.
Repeat the question.
So the question was, people or parents who have children are sleep deprived. And I know that for a fact because I'm also a parent. And I actually read that, sleep deprivation would last 6 years until the kid is 6 years age. And my kids are just turned 6 years old, so I thought, oh, I won't be sleep deprived anymore. But obviously, that's wishful thinking. So the question was, do parents with children compared to people who don't have children, are they more susceptible to Alzheimer's? So it's actually a good question that no one has answered at this point. It would be actually great to actually look at the susceptibility of patients who are known Alzheimer's patients, who were parents, who were sleep deprived when they were raising their kids, versus those who did not have children. So no one's done that research as far as I know. But I think that would be a good research question that could be or should be addressed.
Are you saying that children derive
there's no association right now. If you have children versus you don't have children.
Better not.
I hope not. I hope not too. Yes.
Is there a difference between being normal, going to sleep and sleeping, and having to take a sleeping pill and sleep?
As far as current research has shown, there's no difference. So if you do need sleep aids, so actually my next slide is what strategies can we do to make sure that we have normal sleep. So the ideal sleep is probably 8 hours. But again, nowadays, we can't sleep for 8 hours. And sometimes, at our age, middle age, it's hard for us to have a normal, sleep the whole 8 hours. But we have certain strategies here. So exercise is one of those. There's a study where people who who do exercise during the day tend to sleep longer. all right? Probably because they're tired. So that's one thing. And don't take caffeinated drinks. So that's a very good strategy. Some people are even more susceptible. So if they drink coffee or any caffeinated drink in the afternoon, it's almost rest assured that that person could not sleep in the evening. So if you're that kind of person, so don't do that. And treat the, but sorry?
Or chocolate.
Oh, yeah, or chocolate. So I put food in there. So chocolate is obviously one of those caffeinated items that you could take. Treat the bedroom as bedroom. So because the bedroom is bedroom, so don't work in your bedroom. Don't do any activity that is not sleep related. And make the bedroom dark. So some people, for example, myself, I could, if I detect a light, I wake up right away, straight away. So make your bedroom dark. So invest in a light-proof curtain if you need to. Yeah.
You can wear a sleeping mask like I do, so when the sun comes up, it doesn't bother me.
Yeah. So if that's even better, if some people are not susceptible to light. So if you're that kind of person, then do sleep longer. All right. Handle stress. So stress has been shown to actually influence cognition and learning and memory. So short term, it's fine, but long term, it's usually bad. So cortisol is one of those molecules that is released by our body to cope with stress. However, too much cortisol actually influences, specifically affects brain cells in the hippocampus. Yeah.
Define what kinds of long-term stress are there?
Yeah, so long-term stress is basically a stress that spans months.
Well, I mean, what causes? Examples of it?
So work could be one reason, but I guess for many of us here in this room, work is not an issue anymore. It's not a source of stress.
What?
Work. So, or business. So anything that basically triggers stress long term. So, I can, say from my experience, being a professor is stressful. So I look forward to my retirement. But there's obviously benefits for being a professor. In terms of, I guess, stress, in terms of like family, based stress, like if you have a family member that's giving you stress, then try to basically get out of that environment. So how do you handle stress? Again, you'll probably find exercise and some of these strategies kind of overlap. Relaxation techniques, meditation, some of you might be doing that now, mindfulness. I find that doing yoga helps me a lot. I do the hot yoga, Bikram yoga, and do that 3 or 4 times a week. Sleeping, obviously, also helps in relieving stress. Socializing also, minimizes stress. The diet, and again, so changing your environment. So if you're in a stressful environment, as much as possible, you should avoid that environment. Interact. So interact or be socialized. So right now, what we have here is a group. So this is one way of interacting and socializing. So coming to lectures like this, post-lecture, having activities within the OLLI group, it's very good. So there's been a suggestion that friends make you smart. So having that interaction, so there's a research, at least it's associated, that interacting with your friends, socializing, actually enhances your cognition, your memory, and obviously your learning as well. So strategies, spending time with families or friends. I put here, joining interest groups such as OLLI, arts and crafts, and also volunteering at organizations, nonprofits. So that's one way for you to socialize and interact with other people. Exercise. So physical exercise has been shown to actually improve not only mood, and not only physical, cardiovascular, your heart, but also your brain as well. They actually performed studies in animals where they have, again, rodents are typical animal models. So they had rodents that are model of Alzheimer's disease. They have rodents that are normal. And they have rodents that are Alzheimer's that are not exercising. So they compared the brains of those, and they found that the rodent models of Alzheimer's disease, they have less plaque accumulation in the brain when they looked at it. For those who have exercised a lot, like daily, they put those little wheels, compared to those who did not, basically just kind of sedentary lifestyle equivalent of humans. And this actually study just came out yesterday. So I just put there. So new neurons actually are born in your brain, particularly in the hippocampus, when you exercise. All right? So there's actually a controversy last year. So in the past, I think almost 15 years ago, Fred Gage from San Diego produced a study that showed, adults actually have new neurons that are being born. Last year, there's a study that actually contradicted that. They said, well, we looked at these brains, and none of these brains showed any new neurons. All right, so that controversy basically triggered this new study that got released yesterday, that in fact, when they, the difference was the methodology. So that the other lab that published their findings last year used a different method, and now what they did was they tried to compare their method and now a new method that they developed that actually showed that in fact up to 90s, in our 90s, we have still new neurons that are being born in the hippocampus. So exercise is one of those triggers that create these new neurons. So what strategies can we do? Well, again, walk instead of driving, and if you are not able, do something physical if you can. Again, if you're able, you could join a local gym. Some gyms have exercise groups where you could actually become motivated in doing the exercise when you're in a group compared to you're doing it by yourself. Physical activities at home, like gardening, and anything that makes you move in your home will also help, and that will increase your basically your heart pumping and basically getting all these blood into your brain. Learning new things. So hopefully today you learn new things, like the brain cells, and so that's one of those proposals or hypotheses where they actually showed that if you learn new things, again, learning is making sure that your memory is intact. You're having the ability to remember things when you actually try to recall. So when you learn things, you stimulate these neurons in your brain to actually form new connections. And these forming of new connections actually constitute the memory themselves. Right? So it allows you to learn more things. So strategies, again, similar to what I mentioned, join a local group. You could even, take a course at the university or community college. There's actually a news that I came across. A person who got diagnosed with Alzheimer's actually started to study at a university. So this is in Canada. That was a month or so ago. And according to this person, obviously it's not based on scientific studies, that he actually, he found that the challenge of learning something in a class environment, within the university setting, allowed him to actually have, a better memory. So learning a new language might be, I guess, unless of course you're moving to a new country, you don't necessarily need or want to learn a new language, but it's one of those. Learning how to play a musical instrument. So when you were young, I remember when I was young, I started learning piano, but in the end I got bored and I stopped, playing the piano. But it's something that, if you started something when you were young, musical instrument, maybe that's something that you could start doing again, since you have plenty of time, to actually do things now. And then do activities that challenge your mind, puzzles. And also there's a recent trend where merely getting a book, coloring book, and coloring actually improve cognition and memory. So that's a recent trend that if you have not heard of, something that you could start doing.
What's Kumon?
Kumon is basically those numbers where you try to kind of fill the numbers, or it's like a puzzle. Diet. So you are what you eat. I don't know who actually said is it Anthony Bourdain? You are what you eat? But it's one of those shows that you are what you eat. But anyway, so recent studies suggest that the Mediterranean diet, which is plant-based, legumes, whole grains, good oils, olive oil, actually help cognition and memory. And this is from the NIH that actually shows that there's a study. They looked at association in diets, and they found that the Mediterranean diet actually slows the progression of Alzheimer's disease. So this is something that you could start now. Some of you probably love your meat, but it's not too late. You could still change. You could supplement instead of using like peanut oil or canola oil or something. Maybe use olive oil. Good fats. For snack, you could probably have walnuts. I don't know if you noticed, walnuts are like almost the shape of a brain. All these folds. And indeed, actually, walnut is supposed to be good for our brain because it contains the good fats that the brain needs. And strategies I've mentioned, so eat green leafy vegetables if you are able to, salads, oily fish, salmon, it's one of those main popular oily fish, fruits and vegetables. So blueberry is actually one of the, what they call the miracle berries. Supposedly, again, based on some studies, that it could actually help, learning and cognition, the brain.
Which one is that?
Blueberry. But almost all berries are actually, they have these, good antioxidant molecules. And again, socializing. So enjoy a dine out with friends or family. It's something that you could do as a strategy for diet. All right, so we're getting close to the end. One last thing. So I mentioned to you about SHIELD, but I'm adding O. All right? And this is based on a very recent study as well. So oral health. So a few months ago, so this is Rudy Tanzi, my former director, and this is actually Rob Moore. He's a former colleague as well at Harvard Medical School and Mass General Hospital. So he's been actually focused on looking at infectious material that have been found in the brain of Alzheimer's patients. All right? And there's a recent study that actually found the bacteria that causes gum disease, the P. gingivalis bacteria. The bacteria produce protein, and these proteins are toxins called gingipains. All right, the gingipain is actually toxic to neurons, in your brain, and they found it in Alzheimer's disease that the toxins are actually in the brain of Alzheimer's patients. So oral health is therefore also important. So strategy would be brush and floss daily if you can, mouthwash, go to your dentist regularly, and eating healthy again, which is an overlap, in one of the strategies for SHIELD. All right, to summarize, the brain is very complicated organ in our body. There's a lot of research that's ongoing, and unfortunately, Alzheimer's disease ravages certain parts of our brain. One way to probably protect ourselves is to shield ourselves, all right, in order to hopefully, protect or avoid or minimize ourselves from suffering with Alzheimer's disease.
What's
Yes.
the O stand for?
Oral health. All right, so I just quickly want to acknowledge I'm funded by the National Institute of Health. These are current researchers in my lab, and a few have other students in the lab as well. So thank you very much. If you have any questions. Yeah.
Maybe we have time for one question.
Talk about memory. Is there any research that tries to look at the distinction between what you remember and your ability to recall it? Because
So there is a difference. So so in Alzheimer's patients, the ability to recall and actually remember are impaired. But some form of normal aging is just failure to recall. So that does not mean that person suffers from Alzheimer's. So the recall is, I guess, it could be due to some other factors like, again, stress, lack of sleep. That influences our ability to recall, but that does not mean that you're actually going to suffer from Alzheimer's or suffering from Alzheimer's disease.
When I think of it is, I can't remember something. This has always been true. Mm-hmm. But as soon as I say it, oh yes, I remember.
Yes, yeah. So that is more of say a pathway of how a certain neuron gets activated, and you're trying to recall, but the connection is not quite active or strong, and therefore you could not recall the memory. But then there's a trigger, like for example, seeing something which activates another neuron that will then kind of trigger that pathway to say, oh, now that memory is now active, and therefore you recall. So that's sort of again, it's somewhat normal, especially as we age. That's something that happens. It's because, again, our brain is very dynamic. The neurons usually change connections all the time. So today, your brain changed because you learned something new. That connection, again, changed. It connected to some other parts of the neuron, and some of the connections may be retracted a little bit because of the change that happened today. Yeah, these connections.
Thank you so much.